Hello everyone. So I will talk today of the use of supervised machine learning methods to infer demographic history. And first, a few words about demographic history. By that, I refer to historical population dynamics, like past events, past changes that happened through time. And in the case of humans, for instance, we are interested in how we colonize different parts of the world. And in particular, if we look at now current populations at the present time, we are interested in reconstructing some past events, such as when population separated in the past, or migration rates between these populations, or effective population sizes. And so we can do that thanks to genomic data, because genetic variation is shaped by past demography. As a well-known example, I can mention the founder effects, in which we have some individuals coming from a larger population that is founding a new population that leads to a decrease of genetic variation. Something that we can see also with bottlenecks. I can mention also that when we have populations in expansion, we can see that we have much more rare alleles than a population with constant effective population sizes. Another example, when we have looking at the length of some haplotypes, we can date past events such as migration. And then for doing demographic inference, so what we will do is that we will sample individuals coming from different populations that have undergone complex demographic histories. And then from the genomic data, we will first specify a demographic model that is a simplification of what we can see for real. And then we will estimate the demographic parameters from this population, such as the one that I described before. And this is this second task that we will tackle to estimate this kind of parameters for interacting populations. And we will do that using simulations to train predictive models. So we talked a lot about simulations, the usefulness, the difficulties also. So I can mention that it first developed strongly in demographic inference with approximate Bayesian computation, in which we estimate a posterior distribution of demographic parameters by comparing summary statistics from simulated genomic data and real data. And then there were methods using directly the simulation to train a predictive model, such as, for instance, for predicting effective population size with a neural network in the seminal paper Deep Learning for Population Genetic Inference from Sarah, and some random forest for classification or regression task in population genetics. And this is this kind of approach that that we use in our case for our demographic situation. I show here the global process. I will come back on it later. So first we simulate our genomic data, then we compute summary statistics on them. Then we can use directly regression methods on these summary statistics. And then we have, of course, to evaluate how this kind of regression methods perform. So first about simulations. So we simulated genomic data of 10 individuals coming from a population 1 and 10 individuals coming from a population 2 that separated at a given moment in the past, coming from an ancestral population. So this is what we call an isolation with migration model. We simulated genomic data thanks to MS Prime, we simulated 20 loci of 2 megabases using average mutation and recombination rate for humans. And so we simulated 10,000 scenarios, so 10,000 set of genomic data, sampling randomly the demographic parameters in uniformly from 1 to 5,000 generations for the split time from 0 to 1/1,000 for the migration rate, and from 100 to 10,000 for the effective population sizes. And so this is these parameters that we want to infer. So then the idea is to compute many summary statistics. It is the advantage of this kind of using this regression method over ABC, for instance. And so the idea is that we compute many different summary statistics, and we will let the regression methods use the ones that are useful. So first, we computed summary statistics at the scale of population. In that case, we computed it on population 1, population 2, and the overall population. Here I show the classes of summary statistics. And I say classes because actually, for instance, in the SFS, we will have many features in our training dataset. We will consider as one feature the percentage of SNPs that are derived one time or two times, and so on. And we will also consider the distances between the SNPs that have the same frequency. For the linkage disequilibrium, we consider the correlations between SNP over different set of distances. We consider also the length of segments that are identical for 2, 4, 8, or 16 haplotypes, and the AF-IBS that is also considering the length of identical segments, but this time looking at the SNPs that have the same frequency. And we also consider multi-population statistics. So first, the joint site frequency spectrum, in which we will consider all combination of derived allele. For instance, one feature will be the percentage of SNPs that is derived 3 times in population 1 and 5 or 6 times in population 2. And two statistics that are more summarized, the FST and the divergence, that summarize genetic differentiation between our population. And so here I am showing the different features that we will have in our training set coming from these classes of summary statistics, so many different features given the classes. Then the regression methods that we explored on these summary statistics. So first, the random forest that was quite used for a population genetics task. The XGBoost that is also a tree-based model, an ensemble model, and a neural network, the multi-layer perceptron. And so we trained these regression methods on our training set of summary statistics. And then we evaluated how these regression methods performed to predict the demographic parameters thanks to metrics comparing the observed values and the predicted values that I will show directly here. So we have the observed values on x-axis, on the x-axis, the predicted values on the y-axis for all 3 regression methods, random forest, XGBoost, and multi-layer perceptron, and for the demographic models. So one thing that we saw, that is all these models were able to achieving good accuracy in predicting the demographic parameters with some differences. For instance, the ancestral effective population size was demographic parameter predicted the most accurately by all the methods. So the accuracy very close for all these 3 methods. But for the other demographic parameters, actually we have differences. With the MLP performed better ahead the XGBoost and ahead the random forest. So I show this looking at the metrics. And why we have that? For instance, for the split time, we can say that we have a slight bias for high values of separation time for the random forest and the XGBoost. Then, once we saw the performances of these regression methods, we wanted to see what were the classes of summary statistics used by these regression methods. And I show that here for the split time, random forest, XGBoost, MLP. And so here we have our classes of summary statistics, and the bars are represented the importances computed with the permutation feature importance method for the different regression models. And so for the random forest and the XGBoost, what we can see is that even though the XGBoost performed a bit better than the random forest, actually they used quite the same classes of summary statistics. In particular for this demographic parameter, the side frequency spectrum. But for the MLP, it was quite, we saw that it used quite different classes of summary statistics with the joint SFS coming ahead the AF, IBS, and SF side frequency spectrum classes. And it is something actually that we saw for the other demographic parameters, the ancestral effective population size, the migration rate, because we have the FST that was used with high importance with random forest and XGBoost and not by the MLP, and the current effective population size. And so then we wanted to see more precisely what was the summary statistics, the unique features used by the different regression methods. So not at the classes scale, but really at the feature scale. And for the random forest, actually looking at the top 5 features among these many redundant features that we wanted to give to the models, we can see that the top features are achieving a high importance. For instance, for the split time, the random forest use this feature that is the number of tripletons in the overall population with an importance of more than 20%. And it's also the case with the other demographic parameters. We have feature with high importance. And it's also something that we have in the XGBoost, with the XGBoost, but not with the MLP, for which the top features are just achieving a few percent of importance computing with permutation feature. So what we can see here is that with these two methods, we are able to detect very important features for the prediction, while for the MLP, it's not the case. And looking at more broadly, the top 50 features look for all the regression methods and representing here by warmer colors, higher importances, and with colder colors, lower importances. We see actually that for the 2 tree-based methods, random forest and the XGBoost, we have warmer colors, while the MLP is using much more intermediate importances for the prediction. That implies that for its predictions, the MLP method involved more complex summary statistic relationships, spreading the importances on many different features. So until there, we looked at the importances of for the different regression method, but as we talked in the previous talk, because with this kind of method, we don't have linear effects, so we don't have straightforward interpretation of these of the effects of summary statistics on the prediction of the demographic parameter. And it is something that we tackled here using the method called SHAP that quantify the effect of a given summary statistic on the value of a given summary statistics for the prediction of the demographic parameter. And it was in particular something already used in population genetics, in relation with recombination rate by Berger et al. And so here I show the results for the prediction of the split time by XGB model. That was a model that was able to detect some very important features. And so one first thing that we saw is that we have the same top feature that we get with the permutation feature importance, with the number of tripletons in the overall population as very important in the prediction. And then thanks to SHAP values, we can see the impact of these features on the prediction of the split time. When we have weight points, that mean high value for the feature, that mean high number of tripletons in the overall population, we have high SHAP values. So that means that when we have high number of tripletons, we tend to have more higher separation time between our populations. And so as a conclusion, so with this work, we were able to see that using this kind of supervised machine learning methods based on a large number of summary statistics, we were able to predict accurately demographic parameters for interacting populations. We were able to distinguish some differences in the prediction performance with the MLP that performed best. We were able to see that this kind of regression methods use different classes of summary statistics. And we were able also to, using interpretability method, to see how these summary statistics can influence the prediction. And then as future work, we will want to see how it can perform some deep learning methods using directly raw genomic data, if we can outperform what we did with the method using supervised machine learning method on summary statistics. And so I would like to thank you all for your attention, and as well my supervisors and my colleagues. Thank you.